And the antigen antibody interaction. How this antigen antibody interaction take place? The antigen antibody interaction take place through the use of the many of the bonds such as the hydrogen bond, electrostatic bond, hydrophobic interaction, and the van der Waals forces. And these antigen antibody interaction is the non-covalent and reversible interaction. This is very important to use the antibody in the various other things also. After antigen binding, the constant Fc region of the antibody performs the effector functions, and the effector functions include the antibodies defend the organism by a number of effector mechanisms, and these important mechanisms can be a neutralization, opsonization, or the complement activation. What is this then? Neutralization. Neutralization is the uh, antibodies code the antigens present on the surface of pathogen and prevent their entry into the cell. And these antibodies are also code the toxin released by the pathogens and prevent their toxic effect. Here the neutralizing process is taking place. These antibodies are interacting on the antigens and on their these epitopes and don't allow any cells to any these antigens to enter through the cells, and they are able to distinguish between these antibodies, these foreign pathogens, and from the our own cells. This is self-recognition. Then the opsonization. Opsonization is the antibodies coat the surface of the pathogen and they facilitates the phagocytosis. Okay, they facilitating the phagocytosis by attracting the phagocytos. These cells, when interact with these uh, antigens, release some of the enzymes such as grand enzyme and kill these microbes by creating a pore or dissolving their cell membrane from their surface and killing those the pathogens. And these antibodies also attract the natural killer cells, and the natural killer cells causes the antibody dependent cell mediated cytotoxicity, which is called as the ADCC, which kills the pathogens. And the complement activation. During the complement activation, binding of antibodies to antigens on a surface of pathogen can activate the complement system. And this complement system either destroy the pathogen directly or facilitates their destruction by the phagocytome. Here the complement activation, here these complement proteins are activated and they are phagocytes are placed and they are killing those microbes.